second section here, we're going to look at actual chordates. So those are the hemichordates, which resemble chordates in some ways more than the other echinoderms do, I would say, but are actually phenotypically, phylogenetically a little closer to the Ambulacraria, to the echinoderms and creatures like that. And so the chordates are over on the other branch, and these are going to be more like us. We belong to this group. So let's look at them and let's turn on those subtitles. There we are. All right, so there's going to be 5 main features that define chordates. And these features are going to be present at some point during chordate development. Now, the real way that we define chordates is by genetics. So even if you were to pull a chordate out of the ocean and it was missing, 3 of these 5 characteristics, which would be weird. If genetics told you that it belongs with the chordates, then it is going to be a chordate, and yeah, we can't really argue about that. The genes basically trump anything else. So the 5 characteristics are going to be a dorsal nerve cord. And so in humans, this is going to be the spinal cord, but it's just going to be a bundle of nerves that are essentially an extension of the brain. So the brain's going to be up here, and the dorsal nerve cord is going to stretch down the back, and it's going to provide nerves to the rest of the body. And they're going to branch out sideways from this. And if you've had A and P, you know the different nerves that branch out at different points along the backbone. Now, underneath the dorsal nerve cord, which is located right on the back, but underneath it is going to be the notochord, which is meant, or is kind of a stiff rod. And so in some cases, this is going to be cartilage. In our case, this is actually going to be the spinal cord. So the spinal cord runs on the inside. The actual, the vertebrae run on the inside. The spinal cord is on the outside, and it's actually running through little holes. Our spinal cord has little holes that are going to wrap around this to try and protect it a bit more. But if you were out to design a really good animal, I just don't see why you would want to put the dorsal nerve cord, which, controls all the limbs, and if it gets damaged, you're paralyzed, on the outside, and put the thing that's there to protect it on the inside. I think the answer is this is just the way it evolved, and you don't get a choice about these things, but there you go. So in addition to that, we're going to have pharyngeal gill slits, and these disappear in adult humans. Our gills actually mineralize, and so we're going to turn some of these into ribs that are going to protect our lungs. And so the organs on the inside that we're actually doing the exchange of gases, those are going to still be our lungs in the adult. But humans go through a couple really neat metamorphoses, quite at least as interesting as what you see in insects. And yeah, very striking changes. Anyway, the top one of these little pharyngeal gill slits is actually going to mineralize up, and that forms the lower jaw. So you're it's kind of neat. The top of your skull actually forms around the brain, and that's going to form separately from the lower jaw. And that's going to migrate up later during development. We think this was also the evolutionary pattern, that jaws come from essentially the top rib becomes a jaw. So that's neat. But anyway, yeah, those pharyngeal gill slits located in the pharynx area, and that's where we're going to exchange gases. So that's going to be fine. The next feature is going to be an endostyle. So this is a groove lined with tiny hair-like cilia that transport food. This is how a lot of cephalochordates will still eat. It's a feeding device that they use. In the other members of the chordata, this is going to tend to move down inside. So in humans, this is going to end up down in your throat. It's going to be some of those cilia that move mucus along are going to derive from our embryonic endostyle. But it used to be a little tentacle mouth. So yeah, little tentacled babies. Yeah, how cute, right? And in some of the other chordates, we're going to see the endostyle move down in, and it's going to end up also around the pharynx. But I'll show you pictures of that. That's all coming up. And then the last feature of the big 5 is going to be the post-anal tail. And yeah, in our case, it's only in the embryo, but a lot of other things, think like fish or reptiles or, yeah, even birds would have a tail, not the part with the feathers, the part that's underneath the feathers. And a lot of mammals. Dogs and cats are still going to keep their tails, but humans have lost them. We are a weird tailless mammal. But we do still have that tailbone that's going to be tucked away, and that's going to contain the remnants from the embryonic postanal tail. We just keep it tucked up inside. So here are some of the larvae of different members of this group. So shown down at the bottom, we have tunicate larva, and we have lamprey larva, and we have, that bottom is a cephalochordate. But it has a really nice way of showing the little parts. So imagine these two are the same. So here we've got the endostyle up front. So that should be, the little oral tentacles. That's fine. That's what we're going to use to eat. And you can call them oral tentacles or an endostyle. That's fine. Down in here, this is going to be the pharynx, this sort of brown thing that looks like a harp. You're going to have the brain up here with or without eyes. But if eyes are present, they're going to be an extension of the brain. And so, we have to remember that our eyes are actually that one little part of the brain that pokes out through two holes in the front of your face. And lets us see. So it's a useful kind of thing, but still kind of weird. Then here's going to be the brain, and then this is going to be the dorsal nerve cord. So that's presumably, D is the medulla part. And so there we go. B is the dorsal nerve cord, and it is this blue thing, and it's contiguous with the rest of the brain. The thing underneath that is going to be the notochord, and so that's down here in C, and then we're going to have a little gut down in here, which is going to be our P and Q, and the anus is going to be right there. It's not marked, and some of these guys will have a little sort of extension of skin, which is like a tail, which helps them to swim a little bit better. And others don't have this. And so baby humans look like this. Baby lamprey, which is a kind of a primitive fish that doesn't have any jaws, look like this. And baby tunicates also look like this. So here's the little tunicate notochord. Here's the tunicate nerve cord, tunicate brain. And these are the other organs of the tunicate. This is the pharynx down in here, this little sort of bag with the slits in it, those four little slits. And we will show you all those things in much more detail. All right. So comparing the chordates to the hemichordates, we actually have a lot of genes in common, and genetics is the best way to actually look at this. And so shown here are the areas in the embryo, and we actually get the same genes, and they're turned on in the same places. And so that kind of tells you what the equivalent parts of the body are. So the face, if you like, and the eye are going to be produced by the same sort of genes that produce the proboscis of the enteronucleus, which is a hemichordate, pardon me. The back of the brain, sort of midbrain and medulla back here, are going to be, actually no, just the medulla, and the first one of these gill slits are going to be controlled by some of these genes. So there's a PAX gene, and we know about PAX genes, and you'll have other things in here like Hox genes. They are very important in development of the extremities, like arms and legs, for example, in humans. Same program is going to operate this part. So the collar and the first one of those pharyngeal gill slits, that's all going to be controlled by those same genes. And then the hindbrain, so here's our medulla equivalent, and just going to merge seamlessly into the spinal cord. That is going to be that, and the rest of the gill slits are going to be controlled by the same genes that control that same part in the enteronucleus. And then you get the tail, which is going to be under its own control. Hox 11 and 13 are going to run that for both of us. So yeah, we're basically the same thing. And there are just a few differences, but there are more similarities in the way chordates and hemichordates develop than differences, I would say, at least on a genetic level. So let's talk tunicates. All right, here are some. These little blue guys right over here. Here are some of the same little blue guys. I think that might be a different species with that yellow ring around it. Either that or just a better picture. And these are also going to be tunicata as well. These little sort of green and brown guys down here. And you're going to notice each one of these has two little holes in the top. One is going to be for pulling water in. The other is going to be for pushing water out. So they're going to filter. And as they filter, they're going to breathe the oxygen. So they're going to exchange gases. And they're going to siphon little bits of detritus or little tiny organisms out of the water and eat those. So they're just going to be constantly pumping water, and while they're doing that, they can get food and they can oxygenate themselves, which is very nice. Here is what some of the adults look like, and there are actually a lot of different forms here. So doliolids look like that. They're like a little sort of a rectangle with a little tail quite often. Pyrosomes are going to form big long chains, and so this is a pyrosome chain, and sometimes they'll form big long tubes, but here are some individuals, and you can see those little slits inside. That's going to be the gill slits, I believe. Appendicularia looks like a little larvation, so it looks like a little larval human being or something, and it's going to form, all of this stuff is a giant mucus house. And we got a couple videos that show these things making and using their mucus house. This is really interesting. They make a new one every couple of days, and they're going to use it to filter feeds. They're going to use it to funnel particles into their mouths, and this is basically a tool that these creatures are able to build. So it's definitely not just humans and chimpanzees and other vertebrates that use tools. Appendicularians secrete their own tool. Use it, and when they're done with it, they cut it off, and then they make another one. And then this is going to be the more typical sort of Ascidian. This is an Ascidian that is solitary, lives all by itself. Over here is a colonial Ascidian. They're quite neat. And then salps are also going to be, they're going to look similar to, I think, doliolids, but they're also going to form big long chains. And so these are individuals. The blue bits pointing to the top, the red bits pointing to the bottom. Organs are clustered down at the bottom. Water flow is going to be up at the top. And in something like a salp, there are going to be specialized units. So it's going to be a whole family of related individuals. But some of these units are going to mainly feed. Others of these units are going to mainly power and sort of push, help move the colony along. And they're going to share food, and they are all going to work together. It's very neat. And so here are some more of those tunicates, and aren't they gorgeous? Just really neat looking. So here are two different kinds of adult tunicates. And I would suspect these little bars here are going to be the pharynx, but we'd have to look inside of one to make absolutely certain of that. And here are the two big holes in there where the water comes in and water comes out, respectively. Up here at the top, or at the bottom, sorry, at the top of that thing, we're going to see the notochords. That's going to be running through the middle. The central nervous system is here. So this is the brain essentially wrapping around here and stretching out over the back. And so this is going to be the dorsal nerve cord. This is the head area on a very early stage larva. These things on the inside are actually going to develop into all of its internal organs in time, but it's still at an early enough stage that it doesn't have that. And one of the first organs it does develop is called an otolith. And an otolith is going to be equivalent to one of the structures in the vertebrate ear. It's actually going to be used for balance. And so, yeah, they have a little tiny balance organ right built into that primitive brain, which is very interesting. And so tunicates start out looking very much like a normal embryo. They have all 5 of those features. They have a little pharynx with an endostyle on it. They have a dorsal nerve cord and a notochord, and they have a muscles keep going, post-anal tail for sure. And so what they're going to do is the larva is going to come in and attach. It's got an attachment gland down here, this adhesive surface, and it's going to find a good place and just settle down and glue itself down to something hard usually. And from that point, it's going to live in that spot. It undergoes a marvelous metamorphosis. So watch the internal organs. They're going to start out clustered up here in the head, essentially, head area. So it's actually more like the entire body area, I would say, but it's got the brain wrapping around it. So yeah, I guess maybe that's the body and it has a really long tail. Maybe that's the way we should think about this. But we're going to start out. We used to have a pharynx, which was able to pull in water independently. So that was one. And a second hole, which was the mouth. And watch these things rotate. And so they're going to start moving up. So here's the early stage, and we're going to dissolve those notochord and get rid of some parts of the nerve cord, but keep other parts. And we're going to retain a little tiny brain, but it's just going to be here and a little blob right about there. So there's our little tiny brain now located up there. And then finally, we've rotated all the way around. And so the oral opening, we're going to pull stuff in. we're going to catch the food with the stomach, essentially, first. And then we're going to process out waste down here and dump it back into this chamber. And this chamber is where the water is going to go after, and we're going to pass the water over there, oxygenate all of the rest of the organism, and then the water is going to pass out through the pharynx on its way. So water goes in through the esophagus, if you like, and out through the pharynx opening. And there are the little gill slits, like we saw on the exterior of the larva, but they're now going to be moved to the interior. So it's still got some of those parts, missing other parts, but this kind of shows you where they are. Here's a better, kind of more detailed version of this. And so we are going to have in this one our, let's see, oral siphon with little oral tentacles here. And so these are going to be derived from the endostyle, but the main endostyle is going to be in here. And so the endostyle is going to be this layer. We're going to have a little ciliated layer right here around the pharynx. And so we're catching food with this and breathing with the rest of the pharynx. That's just there for with lots of little gill slits for maximum oxygen exchange. All the food that's processed here is going to be fed down into here, which is going to be the stomach. And it's going to be digested exactly like a stomach would. And then it's going to come through here. This is effectively the intestine. And the anus is going to be in here. And this is going to be where the water flows back out. In this particular individual, this is a sea squirt. It's going to be a little different than some of the others. There are a lot of different architectures in the tunicates, at least superficially different. Located up at the top, you're going to have a little cerebral ganglion, which is going to be the equivalent of the brain. This is what happened to the brain of the embryo. It sort of stayed in place. And then a neural gland right over here, which is going to be the gland hooked up to the brain. And there's also going to be some nerve that runs right down here and goes down to the digestive organs and reproductive organs down here at the bottom. And these guys have different schedules when they will spawn and release either sperm or eggs. So looking a little closer at those gills, so this is the pharynx, and you can see these are the blood vessels that are running there. And look how these are maximizing the surface area for gas exchange. So the blood vessel is going to be flattened out like this, and there's our sort of trunk. This is the vessel that's connected. And they're going to be arranged in something like a waffle pattern, so crisscross. And yeah, these are the little veins that are going to be allowing gas exchange on the inside. So this is what the pharynx is made of. And this is why it has that appearance. The waffle-like appearance goes right down to the microscopic level. And there are going to be little holes that allow water access into these areas where the blood vessels are. And that is effectively going to be the gill. That's how they're going to exchange gases. Shown down here, let's see. This is, I thought these were brains. Yes, okay. So we're looking for serotonin throughout the body. And so serotonin is going to be a good marker of brains because that's going to be a neurotransmitter. And I think that's the only real use for serotonin in the body. Although if you find something else, yeah, put it in the test answer. I'm interested to learn. Tubulin, they're going to be these microtubules that run up and down the nerves. This is how the neurotransmitters are transported. And so that's going to be a good marker where you see red and green together. And I think it's going to make... well, yeah, where you see red next to green anyway, that's going to be, the red's going to be the nerve body, and the green is going to be the little vesicles full of serotonin. And so you can see them here. Here are the little serotonins right on there. The blue is just going to be for nuclei, just showing you roughly where they are. And so these are going to be the little nerves. The brain is located right up in here. This is going to be the in current where we bring in the water. Here's going to be where we push out the water, if I'm reading those correctly. Yeah, I think we have a better couple of pictures of the brain coming up. Here we go. So this is zooming in on that little brain. So it's shown just isolated, just floating there. But of course, it wouldn't really work if it was just floating there. It is actually connected up. You have anterior nerves and posterior nerves. And in most kinds of these tunicates, there are going to be 8 main branches that come off the brain. So here we have 2 on the posterior side and 6 on the anterior side for our total of 8. Here we're going to have 2 each on the posterior side, and we're going to have, or rather, no, laterals, yeah, on the side. That's interesting. But the 4 main ones up here. Something like that. The neural architecture is going to depend on the individual species, so they differ a little bit. But that's one good way to tell them apart. And so from this, we can tell that things like appendicularia are genetically probably the most primitive. And so the big difference here between them and the other groups is going to be this little green bit and the light green bit. So this is the dorsal tubercule. So this is going to be something like the brain, and the neural gland duct. So these are going to be these are the parts of that neural gland that should be right there up in what used to be the head region, and they're going to be shifted off to the side here. But otherwise, we're going to have a nice, fairly bilaterally symmetrical brain. And so at each step, so these are the different brains of the different groups that still exist. And generally, we're going to have about 8 primary branches, but it's going to depend on which kind. And some of these branches are going to be larger or smaller, depending on which one we're talking about. Like presence of lateral nerves is something that seems to have developed later. We don't see as much of that here. But they're going back and reconstructing and saying, okay, so if everybody downstream of this point has 2 lateral nerves on each side, then most likely the last common ancestor had 2 lateral nerves and a dorsal tubercle up here and a neural gland duct right down here, because they all have that. And so it's figuring out what the more primitive branches would have been based on what we have in the modern ones that still exist. And so this or this is going to be at one of these 2 branch points. And something like this is the reconstructed brain of the earliest members of the group. And so here's another one, and I like this one a little better. You can see some of the neural tissues in here. And the muscle band. So the muscles are going to be stained up in red, and you can see some of these little muscles running around, crisscrossing and circling around the body. And then these long, these green things are going to be tubulin staining. That's going to include the neurons, but it's going to be a decent marker for where it is. So down here you can see the actual cerebral ganglion, which is, it's equivalent to the brain, the cerebral ganglion. But because they have neural tissues in kind of unusual different areas, that's why it has this unusual name. I think it's a matter of contention whether or not, I don't know if these things would be conscious. I don't know where their consciousness would reside. It's a little tough, but most likely my bet would be the cerebral ganglion. It's fairly brain-like, quite similar to in other invertebrate types. And here's one more of the little brain. And so this is looking at the cerebral ganglion of, I believe, a different kind or else a younger individual. And so you can see the entire cerebral ganglion here, and located right at the center of that, there's going to be our serotonin nerves. And so just like everything else, we're going to have kind of a hind brain that runs on serotonin, and we're going to have a forebrain that runs on various other neurotransmitters. And they have a nice wide range of neurotransmitters, very much like vertebrates or echinoderms. And that's the end of that section.